Colleagues, good afternoon. It's a beautiful afternoon, and it's actually really delighted to welcome you here to the cancer research theme for the afternoon: genomics, nutrition and risk reduction. And it's lovely that we have such expert speakers from the university itself, from UHL and from TUS, from Bowel Cancer Ireland are here with us. We've a keynote speaker, Dr. Paul Brennan, who's the head of Genomics and Epidemiology at the World Health Organization. And I know nothing about science, but what I do know, from my own background, from kind of a legal philosophy perspective, I always refer to this kind of very famous legal philosopher, probably one of the 50 most known legal academics ever in the world in terms of his contribution. And he spent his whole life talking about essential public goods and what they were. And he decided that in the end there was seven essential public goods, and it was the job of law then to protect and scaffold them. And he talked about life itself being an essential public good, each person having an intrinsic value as a human being and the importance of that. And then he also spoke about knowledge exchange as being there was no hierarchy, the seven were all equal, but so he talked about knowledge exchange as well and sociability. And I think that, for me, this is what you're doing here today, the public good of a university engaging in knowledge exchange, generation of ideas, best practice. And so the LDCRC has been in place since 2021, 49 PIs, and that engagement between UL, TUS and University Hospital Limerick. The centre itself is absolutely pioneering, world-class scholars in it, and the combination and the way they approach their work is fascinating, with digital tech, AI, biomedical, and then patient-centred research. And I think the goal in all of this is to improve outcomes for patients in the Midwest region, but also in the country more generally. And when I talked about those public goods, that's such a noble pursuit, and I'll say a little bit more about that in a moment. But having really talented, outstanding scholars, working for this purpose, for life itself and the impact that it can have, is so noble. And as all of you will know, cancer is incredibly prevalent. Affects 1 in 2 people. I think April this month is Bowel Cancer Awareness Month. It's as you know, with bowel cancer, it's one of the leading cases of cancer deaths. 2,500 people diagnosed every year with it. And one of the things that's very worrying is that now 1 in 10 people diagnosed with cancer, bowel cancer, are under 50. So that's a worrying trend. And it's great that we've experts like Paul here who can actually work with this and actually engage through knowledge exchange on this area. And it's to understand the reasons why is this happening, is to problematize it, but it's also then hopefully to prevent it going into the future. And so research in this area is absolutely critical. And the Breakthrough Lab Tour, which I think is the 14th of May in Bernal, is going to kind of assist with this, and particularly how we inform, how we research, and how we empower patients in the Midwest region. And so, just on a personal note, like all of you, or many of you, like most families in Ireland have been impacted by cancer. And I was thinking before I came over this morning, all of my grandparents, my father, my sister, no different to many of you. But I've seen firsthand what science can actually do and what research and the impact that it can actually have. I explained previously to Aideen, my own wife was diagnosed with malignant melanoma and was given 9 months to live. And so she went, it was through science and immunotherapy drugs that she was able to live. And actually, just for us, the esteem in which we hold, she's only alive because of science. And so the esteem that we hold scholars and researchers like you who engage in this pursuit so that other people can live, so that a mom can see her children grow up, has been phenomenal. And actually, even 2 weeks ago, in order to do something in return, the university, CUH, Cork University Hospital, asked her some time ago would she tell her story, and it's part of a story on cancer journeys in St Stephen's Green. And so then a journalist contacted her 2 weeks ago to say, would she tell her story in the Irish Independent, which she did. And it's actually just told it to give others hope, but it's also an acknowledgement in particular of the work of scientists like you and scholars and the medical profession who keep people alive in circumstances which wouldn't otherwise be the case. So delighted to be with you here today and to acknowledge the work that you engage in. Thank you very much.
So I couldn't have done a better introduction, I think, than that. Thank you for those remarks. So in the interest of time, and I think for everyone here to not hear me speak, but to hear our speakers talk, I'll welcome Paul to talk to us today about his work, which spans both international, geographical boundaries, and also spans different cancer types. So he's done a lot of work in colorectal cancer, esophageal cancer, and also renal cancers. A lot of the esophageal cancer cohorts that he's working with are in northern Iran, for example, and then the colorectal cancer cohorts are across Europe and internationally across the world. I think we're extremely grateful to have Paul here to speak to us today, and I'm hoping that we can firm up many collaborations going forward. So with that, I'll welcome Paul to the stage.
Thanks, Kieran. It's a real pleasure to be here. I had a great 24 hours up to now. I had a lovely evening last night chatting with Brendan and John, two patient advocates, and a wonderful morning where we were talking a little bit of science with various folk here. And also just a wonderful tour of the university. It's my first visit to the university, and I found out, it's very impressive, it's very beautiful, and it has an incredibly rich history. So it's just, well, thanks to everybody who's given me their time so far. So I'm from the International Agency for Research on Cancer. It's the research part, the cancer research part of the WHO. We're based in Lyon, in France. I'm going to spend 1 minute, I think, because it's an interesting story about how the agency was initiated 60 years ago, because I think it talks to the power of patient advocacy. And it started with a letter, and a fellow, he just lost his wife to cancer, and he wrote to the local newspaper, a very small little newspaper in the south of France, saying, we've won all these wars, and we've had all this, military expenditure. Why can't we now, win this war against cancer? And that was in the early 60s. And the editor of the newspaper, he knew Charles de Gaulle very well, and he brought the proposition to Charles de Gaulle. I mean, in a couple of weeks, Charles de Gaulle, he'd proposed to the UN to set up an agency for cancer research that would be under the WHO and would stimulate research into prevention and early detection. So I thought it was always wonderful to see how just with one letter from someone who suffered by the loss of his wife, how that can have such a big impact. So I'm going to be talking about this problem where in the last couple of decades now we've realized that in many countries we're seeing this increase in colorectal cancer among young adults. We've been reporting on this now for about 15 years. It was first detected in the US, in Australia, and Canada, and the UK. But now we see it pretty much in every industrialized country. And this at a time where there's been a lot of success with colorectal cancer. So it's among older adults, it's been stable or it's even been decreasing. But we've seen this constant increase now in young adults. And the increase in individuals born in recent decades is up to 4 to 5-fold higher than individuals born in 1960. So there's clearly something important happening here. And we need to figure out, if we want to prevent this, we need to figure out what the underlying cause is. We're starting to see this in Ireland. We think in some countries they're a little bit, the increase has happened maybe a decade or two later, such as in Ireland. We see the same in France, in fact, that the increase is a little bit later, but it is happening. And I think what we're seeing in Ireland, what we're seeing in the UK now, where the increase is really quite substantial, gives an indicator of what we can expect to see in Ireland in the next decade or so. So clearly understanding the reasons behind this and being able to interact or being able to act on that is going to be crucial if we want to prevent this increase in occurrence of colorectal cancer among adults, especially in their 30s and their 40s. So there's been a lot of public awareness about this. There's an increase in concern about what the underlying cause is. Hang on, this one here. There's a lot of academic editorials, calls to action. Most of the focus at the moment is on potential risk factors that we know are important for colorectal cancer. So things like body mass index or diet, highly processed foods, alcohol, metabolic syndrome, etc. The evidence for those at the moment is pretty weak. I will show a little bit of some of the evidence for those risk factors, but I don't think they can explain what's happening. And it's not clear why, if they were important, why they wouldn't be acting at older ages either. And I think the failure to understand what's causing this increase in colorectal cancer among younger adults is indicative about, really our failure in the last couple of decades in identifying new causes of cancer. And I just wanted to spend a couple of minutes talking about that, because if you think about what we know about the causes of cancer, if you think, there are very many international bodies that have reported on this, and basically we can explain about 4 cases out of 10 in a country like Ireland based on known risk factors. Tobacco is still the most important one, with obesity also having a substantial role. CRUK includes healthy diet, eating fruit and vegetables here. Some people would even argue about that, whether the evidence supports a strong role for fruits and vegetables in cancer. But we'll leave it there for the moment. But with all of these, with alcohol, with UV light, with various infections, etc., like papillomavirus, Helicobacter pylori, etc., that have been discovered in the last decades, then we can explain about 4 cases out of 10 that are occurring. Most of these risk factors were actually known like 4 or 5 decades ago. This is the causes of cancer, which was basically the 2 eminent epidemiologists, Richard Doll and Richard Peto from the University of Oxford. They were asked by the US government to write a report on what we knew about the causes of cancer at this time. And it's a wonderful read. You can still pick it up on Amazon. And it's a wonderful introduction to cancer epidemiology. And it's still very much worth reading. And they basically spelled out many of the things that we still see to be true today about cancer. Every cancer that's common in one population is rare somewhere else. There's no cancer that's common everywhere. And there are some cancers that are rare everywhere, but there's no cancer that's common everywhere, which is indicating that cancer is probably preventable. If it's rare in some population and it's common in another, and if we can argue that that's not genetic, and we usually can argue that it's not genetic, then cancer is, in principle, most cancers are preventable. And we can rule out the genetic explanation most of the time because of migrant studies where people move to one country, or because of when there's rapid changes over time. And they estimated that about 8 out of 10 cancers are preventable in principle. And that figure still holds true if you run the numbers today. And at the time, this is Richard Peto, he wrote this in the Times Health Supplement, so you had to dig out to get this. But he wrote there about 30 to 40% of cancer was practicable by preventable means alone, 30% by smoking. And with respect to diet, and I kind of feel this is still true, in the study of diet and cancer anyway, all we know for certain is that we don't know very much for certain. So diet, which has been one of the main focuses for the last 3 or 4 decades, it's still very difficult to pin down what we actually know about diet and cancer. But the most common part was about 30% of that burden was due to tobacco. If you fast forward, this was just published earlier this year. It's from my institute in Lyon, where they repeated this process across the world. And again, 38% of cancers globally were attributable to even a broader list of risk factors. But we still, in 40 years of research, we still haven't really been able to expand the number of cancers that we can explain by known risk factors. The proportion attributable to tobacco has decreased a bit, and a few other things have come into the list, like some viruses. But on the whole, we still can only explain about 4 cases in 10, which means that that one of the arguments I get from this is that epidemiology, which is my background, which is what I've been trying to do for the last couple of decades, is that it hasn't been very successful in recent decades in identifying new causes of cancer, which means we might need to look at other techniques to tease out what the causes of cancer might be. So where are the missing causes? Maybe we're underestimating some things like air pollution. We still think the statistics would say, well, it's probably a very minor contributor to cancer risk. Maybe it's small. It might be due to things that we're all exposed to. And if we're all exposed to it, it's very difficult to identify what the risk is. If everybody is exposed to a carcinogen, then it's just, we won't be able to identify any individuals at risk. Another possible explanation could be windows of susceptibility. Most of the time, we study risk among adults, and often adults in middle and later age. Most of the very large cohort studies of hundreds of thousands of people, they recruit individuals from 40 upwards. But maybe the really important risk windows might be in childhood or adolescence. And there's a lot of genetic studies where we can show that the genetic changes that lead to a cancer often do occur in childhood or adolescence. So there's a strong argument that perhaps there are important windows of susceptibility that we're missing. So let's get back to colorectal cancer. This is something that we were just looking at, a selection of countries. This is from the UK. This is men. You see exactly the same for women. You'll see exactly the same in the US and Canada and Australia, etc. And this is looking at the changes in cancer incidence by these different age groups by year of birth. And overall, if you look at this, you'll see this increase among individuals aged less than 50. But the strong message here is that the increase has been far stronger among very young adults, so individuals in their 20s and 30s. And that, to me, indicates that whatever is causing this increase in early onset colorectal cancer, it has to be occurring before the age of 20. Because we're seeing, even among early, the numbers are modest, but in a large population like the UK, you have enough of these cases to see the trend quite clearly. So the trend is, you see the trend from age 20 onwards. So whatever is causing this increase has to be occurring before the age of 20. So perhaps if we're looking at causes in individuals in their 30s and 40s, it's already too late. The annual percentage change is already high. And in countries like the US, the UK, it's about 3-4% a year. And that's similar to what we saw for lung cancer in the 50s and 60s. It's similar to what we saw for something like oropharynx cancer, the most recent kind of large increase in cancer incidence in the 80s and 90s. And this implies the presence of something that's a strong risk factor for this cancer that is increasing in prevalence. It's the only thing that can explain the trend. So we're looking, either it's a combination of things like obesity and ultra-processed foods, etc., or it's something new that has come in and that we haven't been able to identify up to now. So just a background to all of this, it seems to be a birth cohort effect. It started among individuals in the US, the UK, born around 1960. If you're born in the 70s, your risk is greater than if you're born in the 60s. If you're born in the 80s, it's greater than if you were born in the 70s. So each cohort has an increase in risk of this cancer. Exposure prior to the age of 20, I would argue, seems to be important. So we're not looking at just adult exposures here. We're looking at childhood and adolescence exposures. And the main culprits that we've looked at the moment are things like obesity, diet, like ultra-processed foods, metabolic syndrome, etc. I think at the moment, the evidence is still emerging, and we need to do more on all of these as well. I would say that at the moment, none of these can explain the increase in incidence. This is just a very brief overview of some of the studies. So this was an effort to pull together all the case control studies, looking at some of these risk factors, and splitting them up for colorectal cancer before and after the age of 50. I won't go through all of them, just to say that for none of them was the risk really, apparently a lot stronger for young adults than for older adults. There was this from the Lancet series on ultra-processed foods published last year. Again, ultra-processed foods, maybe they are having some contribution to colorectal cancer, but the risk here in this, which is the third line from the bottom, the increased risk was very minimal, like a 10% increase among individuals having a lot of ultra-processed foods. There were only 4 studies that reported on it. 2 of them showed nothing. 2 of them showed an apparent effect. But again, maybe they are having an effect. But it can't, for me at the moment, these level of effects can't explain the very, very important increases that we're seeing in incidence. So what I would throw out is maybe the cause, the most important cause of early onset colorectal cancer is not due to the known lifestyle risk factors. We need to keep them in mind, but maybe what we're seeing is the emergence of a new exposure, and one that's not captured by our usual epidemiological studies. So this is why I'm introducing this other concept, this other technique that might give us a clue about what might be the underlying cause, especially when we don't know what it is. It's this idea of mutational epidemiology. Basically, if you look at a cancer, if you look at the DNA of a cancer, when you sequence the DNA of a cancer, it's clear that lots of changes have occurred, and many of these changes are due to risk factors. And sometimes now we can link the risk factors to the changes in the DNA. And this gives us a way into understanding what the cause of that cancer might have been by looking at the specific pattern of changes in the DNA and linking that to a specific cancer. So just to spell it out a little bit more, we know the genetic code is made up of adenines and cytosines and guanines and Ts, and we know that these 4 letters, A, C, G, and T, basically spell out all the DNA of every life form, and that there's only 6 different ways when we look at these single-base mutations that each of them can change. So we can have a mutation that goes from C to A, C to G, or C to T, or a mutation that goes from T to A, T to C, or T to G. So we have 6 different base pair mutations. And it's been known for decades that some of these mutations are more likely to be caused by tobacco smoke, or some of these mutations are more likely to be caused by ultraviolet light. I think what the key here was, well, let's expand this a little bit, and let's look at the bases, each pair of the mutations. So there are 16 different ways that a C2A mutation can occur, because you have 4 different possibilities before a base, 4 different possibilities after a base. So if you look across the 6 different types of base pair mutation, you have 96 different types of mutation that can occur, which gives you this mutational signature that we see at the bottom. This is called SBSS1. It's mainly C to T mutations that occur at very specific sites. It's due to an endogenous process that happens in pretty much every cell in our body. And as we get older, pretty much every cell in our body will get this type of mutation. We'll build up this type of mutation as we get older. It's a clockwork mutation, in fact. It's, because the older you are, the more most cells, pretty much every cell in your body will have of this type of SBS1 mutation. There are others that we know are caused by tobacco smoke. So if you look in a lung cancer from a smoker, you'll see a lot of these signature 4, these C2A mutations occurring in this very specific profile. If you look in a melanoma, you'll see nearly all of the mutations will be these very specific types of C to T mutations. And you see other things like aflatoxin, which is a fungus, very important for liver cancer, will be associated. You'll see a lot of the signature 24, alpha-leucic acid, which is a herb, sometimes used in herbal remedies. You'll see signature 20. And then you see other endogenous type processes, DNA type processes. Some of them are caused by well-known chemotherapy type agents. For many of these signatures, we don't know what's causing them. So we're still trying to link many of these signatures, over half of them, to a particular underlying cause. The one I want to focus on today, which is emerging as something that's very important for colorectal cancer and early onset colorectal cancer, is SBS88. And we've been able to understand that this signature, which is primarily these T to C mutations that you can see in the bottom, is caused by a specific bacteria, a specific type of E. coli. And it has this little strand of DNA, to call this PKS island, that causes these specific types of mutations to occur. And this is a study published in Nature in 2020 by the Cleavers group. And you can see down the bottom, they took colorectal organoids, they infected these organoids with this bacteria, they saw the specific pattern of mutations. So they saw that the bacteria was causing this specific mutational pattern. Around the same time, it had just been the year before, colleagues in the Sanger Institute had teased out from the colon of healthy individuals crypts. So our colon is made up of this, the epithelial lining of our colon is made up of millions of these little crypts that each are basically they each come from one stem cell. And you can tease them out, you can whole genome sequence them. And he found that one of the signatures that many of these crypts had was exactly the same signature that had been shown to be caused by this specific pattern of E. coli. So the conclusion from this is that a particular type of E. coli and other bacteria that have this particular strand of DNA, they're causing these mutations in our gut. And perhaps for many of us, it's something that's very common. So these SBS88 mutations, they were also found to be very early events. You could do a phylogenetic analysis on these, and you can see that the events were occurring in the first years of an individual's life. And in that study where they looked in this, some of the individuals were children, and you could see that they had just as many mutations as an adult. And the conclusion was that these are clearly occurring in the first 10 years of life. There was another set of signatures, SBS89, that was also occurring a lot of time, about 10% of the individuals, that they reported they think this is due to a bacteria that we don't know what it is, but we think it's bacterial in origin. So this brings us forward to a study that we just reported on last year, where we were trying to understand colorectal cancer using this mutational signature approach. We were looking across 11 different countries. In fact, we weren't really focusing on early onset colorectal cancer here. It was more trying to understand why some cancers at a higher rate in some regions than other cancers. So for colorectal cancer, we managed to work with groups across 11 countries, recruited about 1,000 individuals with colorectal cancer across these 11 countries. They were all whole genome sequenced. I'm just going to focus on the microsatellite stable cancers here, because the MSI cases, they had so many mutations, you couldn't really tease out the mutational signatures apart from those that are known to be linked to MSI status. And the interesting, surprising result that came from this was that quite a few of the signatures were far more common among individuals with early onset colorectal cancer than later onset. So SBS88 was one that was very clear. ID18 is an indel signature that we've since discovered is also known to be caused by this Colibactin, by this type of E. coli. So these signatures, they're very strongly associated. At least over half of the individuals with colorectal cancer at young age, before the age of 50, had either SBS88 or ID18. So they had strong evidence of, there was a strong increase in these signatures at younger ages. And several other signatures also were associated with young age of onset. There was SBS89, which was previously been reported that we think might be associated with colorectal cancer. It was a lot more common in some countries than others, had particular predominance in South America, in particular Argentina. And there was even another one that we found a little bit everywhere, SBS0. So there was another signature that was emerging associated with young onset. For this, we call it the Colibactin signature because that's the genotoxin that this E. coli produces. It's the Colibactin that makes the mutations. We found it to be more common in high incidence countries than low incidence countries. We found it more common in the left side of the colon and the rectum than the right side. Again, this is very interesting because that's where we think that's where the major increase has been observed for young onset individuals. Other evidence that it might be involved in driving the cancer was that when you look at the most common type of genetic mutation for these cancers, which is mutation in APC, which we know is a very early event in colorectal cancer, you could link those APC mutations to SBS88 or to the indel signature. So we could see that a lot of these mutations in these colorectal cancers were being driven by the Colibactin signature. Just one other aspect of it, when you sequence a tumor, you can pull out the bacterial DNA as well, and you can link that bacterial DNA to a specific bacteria. So these tumors, they were obviously collected at the time when the individual was diagnosed with the cancer, and we didn't see any correlation. There was no association with whether you had the genetic mutation for the signature or you had evidence of the bacteria on your tumor. So it was seeming that whether you were positive or negative for the bacteria at the time of diagnosis didn't seem to be important. It was more whether the mutations were present in your tumor. And the idea for this is that we think those mutations were laid down when you were a child. So the emerging findings from this mutograph study on colorectal cancer, we focused the results on these 800 microsatellite stable cancers from 11 countries, about 100 early onset cancers. So 5 of these were known to be associated with early onset cancer, in particular SBS88, this Colibactin signature that we know is associated with this particular type of E. coli. And a few other signatures as well. And our understanding is that there might be a whole community of bacteria that are causing mutations in the gut. Many of them are causing mutations in the gut in particularly susceptible periods of infancy and childhood. And perhaps these could be, some of them might be more common in some countries than other countries. So SBS89 was very, very common in Argentina, for example. And the way we're following up on this now, one way we're following up on it is looking in the normal colon. So we're going back to the normal colon of individuals. And in the colon, you can tease out individual crypts. So just the emerging picture, early onset colorectal cancer appears to contain multiple signatures. Some are known, like SBS88. We can link to this particular type of E. coli. Some are suspected, like SBS89. The mutations appear to be early events, indicating age of exposure as a key. And the mutation burden can be the equivalent of decades of mutations that would otherwise be occurring at a normal rate. And this is the hypothesis that we want to now test. So we think that changes in birth and neonatal variables and just the way in which childbirth has changed immensely over the last 7-8 decades. Both my parents were born in the west of Ireland in the house where they grew up, without any particular medical care. And I think that was probably representative of many places across the world in the middle of the 20th century. And all of that, of course, has completely changed, and that's been a wonderful phenomenon. But one of the unexpected occurrences of that might have been that we've changed the microbiome of children, and some of these bacteria may be genotoxic, and they're laying down important mutational burdens on children that are given a head start to developing colorectal cancer. And that's the reason behind this new study that just started at the beginning of April, and that's one of the reasons why I'm very happy to be here to talk with Aidan and colleagues, is this new study funded by the European Research Council to understand the causes of the global increase in early onset colorectal cancer. So we're doing this global survey of mutational signatures in cancers across 30-odd countries, trying to understand why these signatures are occurring in children. We're trying to test for the causality of these signatures. So this will involve case control studies where we have representative controls as well to compare the cases to. We're trying to understand the bugs that are causing some of these particular mutations. And then also, I mean, if this hypothesis might be false. It might be, just increasing BMI and ultra-processed foods, etc., or it might be something else. But it might the evidence suggests this could be making an important contribution. And if that is the case, then we need to think of prevention. How can we prevent these bugs? And there might be fairly reasonable ways to prevent exposure to these bugs among kids. And we also need to think about early detection. So if individuals who are in their 20s and 30s now, and if they do have important mutational burdens associated with this, how can we identify them? How can we then prioritize them for screening to make sure that if an individual is at risk of a particular cancer, that can be picked up early and an appropriate treatment can be delivered. So I'd just like to acknowledge various colleagues in IARC who've led this. This has been probably a 5-year work up to now with colleagues in the Sanger Institute, with San Diego, and looking forward to the next steps and hopefully working with Limerick as well on this in the future. Thanks very much.
And I suppose before I get started, I really want to thank the organising committee, and particularly Ruth at 18, for really focusing on colorectal cancer and specifically early onset colorectal cancer here today. As somebody who's at the coalface of telling somebody with early onset colorectal cancer that they have cancer in their 20s or 30s or 40s, it's very, very, very challenging, disappointing, and at times actually upsetting to not be able to tell them why. And I think that's what we're all trying to work towards achieving together. So I think while Paul has just given us such a fantastic overview of the global approach to trying to better understand the etiology of early onset colorectal cancer, I'm going to take a more parochial view. So what I'm going to really focus on is, what does this look like in the Midwest of Ireland? What has it looked like over the past 10 years? So looking at the clinical presentations, some of the social factors, metabolic factors, bringing in a possible concept around accelerated ageing, so looking at the high burden of mutations in such infants and young children, and then the future directions on how we bring this all together. So I don't really need to repeat this after Paul's lecture, but early onset colorectal cancer, it's increasing. It's substantially increasing, particularly in rectal cancer. Whilst there are links to family history, the majority are still sporadic, and ultimately we don't really know why it's happening. The global trends are consistent across the world, and even in Ireland, we started looking at this as far back as 2012 when we could see this data emerging. And if we look in our own department over the past 3 years alone, we can see that the average age of presentation of colorectal cancer has dropped by 3 years, which is quite substantial. But in those 3 years alone, there's been a 33% increase in the presentation of those under the age of 50. So this isn't a concept that's somewhere else. This is here in the Midwest of Ireland. So overall, in the Midwest, we treat colorectal cancer. We diagnose it really throughout all of the hospitals and endoscopy units and radiology units, but the treatment mostly happens at University Hospital Limerick. Annually, we treat about 220, I think it was about 228 last year, surgically treat colorectal cancer overall, and just over 80% will actually be treated with the intention of cure. So they will be stage 1 to stage 3. So in that sense, it's a very nice cancer to work with, but it's that other 20% and it's that younger cohort for whom we don't have that long-term data as well, where it's really, really challenging. So I suppose the aim of putting this data together is when we started talking about how we're going to better understand the etiology, it's first important to understand, well, look, what is this disease entity look like when it's walking through the door of our hospital? So we looked ultimately at the past 10 years, and over that 10-year period, we saw a 100% increase in the presentation of cancers in this group, but actually, this is up to 2024. Last year, we went up to 31, so that was the first time we broke the 30 mark in a year, and we were only one of 8 NCCP colorectal cancer centres in Ireland. We had a full data set in about 114 patients, so that's mostly what I'll go through today. And interestingly, overall with colorectal cancer, probably about 30% of them will be on the rectum, but in this cohort it's 43%. And this particularly is very interesting and really I think where we see the increase arising in our population. So to just look at the clinical phenotype, with colon cancer, we see a left side predominance in particular, the gender balance is around 50-50. Unfortunately, because of young age, these cancers tend to present later. So node positivity is seen in about 2/3. And again, stage 4 is as high as about 23% in the colon cancer cohort as well. And I'll come back to social deprivation at the end and why that's relevant. In the rectal cancer group, it's quite similar, but a much higher rate of node positivity at 80%. So that's 4 out of every 5 rectal cancers diagnosed under 50 are presenting with node positive disease, meaning it's quite advanced at that point. And about 28% of them are presenting with stage 4 disease. So that would be 10% higher than the overall colorectal cancer population that we treat. A third present as an emergency, and that's relevant because if you present with an obstructing cancer, colorectal cancer, you're going to have a worse overall disease-specific and overall survival from that cancer. And again, that, well, we can see if we look at the presentations in colon versus rectum in the first 5 years of this 10-year cohort compared to the second, we can actually see the volume of colon cancers are probably remaining the same, but it's actually the increase in rectal cancers that's really giving us the increase in cancers overall. Then just with regards to some of the treatment, as we would expect, in rectal cancer, we're giving a lot of neoadjuvant treatment. That is increasing in colon cancer as well in younger patients. But I think the frustrating thing is the amount of patients are presenting as emergencies. So they're very symptomatic. These symptoms aren't being considered to be cancer. And then we've lost the opportunity to treat with intention of cure. Recurrence rates are high. Up to 15% will develop recurrence. So this is looking specifically just at stage 1 to stage 3 disease, excluding stage 4. But the recurrence rates are reducing. And interestingly, in contrast to average onset colorectal cancer as well, these patients are far more likely to present with a local recurrence as opposed to a distant recurrence in the liver or lungs. And again, that comes back to probably the locally advanced nature of the cancer at its presentation. Disease-specific mortality, I think the most important thing to look at here is the bottom right of the slide, and that the mortality rate is improving. And I think the reason for that is likely because we are probably getting more aware and probably very marginally detecting things earlier, but also our radiation oncology and medical oncology colleagues and the kind of treatment strategies that they are now, I suppose, very aggressively in, research and trial settings and not just standard clinical practice, making available to patients in this cohort, particularly immunotherapy. I'll touch a little bit on MMR, MSI and oncogene, but Ciara Ryan, who is the hereditary colorectal cancer A&P, who's going to speak after me, will touch on this a lot more and go into a lot more description. But I think the ultimate thing to say from this is we probably as an institution weren't testing this enough and perhaps losing the opportunity to identify hereditary cancer syndromes. But overall, in keeping with what Paul mentioned as well, the majority of these tumours are microsatellite stable. So again, just to kind of make this very specific to Limerick. So social deprivation is a problem in Limerick. We have within our catchment area, we probably have the most affluent area of Ireland and the most socially deprived. And we know that this can impact on your colorectal cancer diagnosis and your overall survival. So when we look in our department over the past 4 years of all colorectal cancers, we know that 1 in 4 will come from an area of social deprivation. And this translates clinically into having a 1.8-fold increased risk of presenting with stage 4 disease, a 3.7-fold increased risk of presenting as an emergency, which we know is obviously bad for overall outcome, a 4.3% increased risk of requiring, or fold risk of requiring a stoma, and a 2.6-fold increased risk of disease recurrence. And essentially all of this translates into worse survival. So And this is work we did with Patrick O'Donnell and one of our previous SPRs and soon-to-be consultant colleagues, Caroline Culnan. And this is really important data for us here from the point of view of trying to identify how to better diagnose and treat patients in these social categories. But interestingly, when we look at this in the early onset group compared to the average onset group, it's actually lower. So social deprivation doesn't seem to be higher in early onset colorectal cancer. It's actually significantly lower in most groups. And that, I suppose, maybe a little bit contrary to what we were thinking, that this is developing because of something in infancy or childhood, but actually from a social determinant, health determinant point of view, then this probably isn't as much of a factor. So again, no closer to understanding the reason why. And then the metabolic phenotype. So I did my PhD on body composition, inflammation and cancer outcomes. So this is something that I always have at the back of my head in cancer in general. But this year, one of the radiology specialist registrars, Anya Daly, did some lovely work on this cohort of 114 patients with early onset colorectal cancer, where she used an AI algorithm and manually measured the visceral fat and muscle areas of all patients at their presenting CT, which is performed in all patients as part of their cancer staging. So we looked at this specifically just in the stage 1 to 3 cancers, because we know stage 4 cancer can really accelerate muscle loss and sarcopenia. So we didn't really want to call something that didn't really exist. So this was 91 patients within that group, and 41% of them had a gender-specific high visceral adiposity rate, and 35% of them were sarcopenic. So then, just to finish, this might be a bit of a conspiracy theory, but I'm going to take the opportunity to talk about it anyway. There are 3 slides. We're constantly trying to understand this, and I really do believe the microbiome, I think Colibactin, I really think we are likely ingesting something environmental with some form of underlying genetic predisposition that is just allowing that activation for these cancers to occur. But when you read too much about this stuff, which is probably the stage I'm at, there's this concept of, well, look, are we actually diagnosing cancers early, or is this a phenomenon where actually, because of all of these exposures, we're more actually aged at that point? So the concept of your biological age or your epigenetic age versus your age by birth. And there's a lot of this work done in the area of dementia and cardiovascular disease in particular. And then if you go further into the rabbit hole, you can find all of these algorithms online where you can put in various factors to try and determine, what is your actual, your biological age versus your age by birth. And if you look at what they use, these are all things we test every day in patients, but it's inherently flawed because all of these things are massively impacted by both physiological and pathological states. They're only really validated in European, white, Caucasian, male and female patients, and they probably report around a kind of standard error of around 3 to 5 years. But anyway, we still looked at it in this population just to see, was this interesting? And actually, I think it is quite fascinating. So what we saw was 80% of this early onset colorectal cancer group had an epigenetic age older than their age by birth, which I thought was fascinating. And then at the last 2 columns, you can see that it's actually increasing in the last 5 years compared to the previous 5 years. And then we stage match the patients with the average onset colorectal cancers. And this is even more fascinating, or else more conspiracy. But you can see from the top row, again, we've got 80% of patients in early onset cancer who have an epigenetic age that's older, but that's only 18% of average onset colorectal cancer. So that kind of, I suppose, there's another rabbit hole that's ongoing from all of that. We did just some correlations between this in the high visceral adiposity group, in your correlating that with your epigenetic age and your chronological age, and seeing a little bit greater correlation in that setting. And again, having high visceral adiposity, we know it's inflammatory, it ages you, and there is a little bit of a kind of a trend emerging there. And then we looked at with social deprivation, and whilst it did correlation both early and average onset colorectal cancer, it was a little bit stronger in the early onset colorectal cancer group. But again, these are just theories still. So, future directions, and I'll finish then. Well, we're absolutely thrilled to have Paul here today. I think the opportunity that he offers us, and particularly patients from the Midwest, to participate in such a global study to understand the etiology of early onset colorectal cancer, but particularly to define that within our population of patients. I think this is just a phenomenal opportunity, and thanks to Aideen for bringing us all together to really take on that. We're just finalising and nearly there with our pipeline for biobanking to take part in the study, and hopefully we'll be able to come back someday and say why this is happening, but most importantly be able to answer that question for patients across the desk when they ask as well. So to conclude, we know early onset colorectal cancer is increasing. The etiology is still elusive. There is a specific phenotype that we see clinically, and we need to match that genetically as well. And I think it creates huge opportunity, and thank you everyone for bringing us all together to really explore these research opportunities as well. So thanks.
Hi everyone. So I'm delighted to be here to talk to you all a little bit about the concept of mainstreaming in cancer genetics and the efforts that are being made to make cancer genetics more accessible. And I'll also talk a little bit about the role of the hereditary colorectal service that we are in the process of establishing over in UHL. I will... broke it. Oh yeah. So I'll start by having just a quick look at the current landscape of cancer genetics in Ireland. So in our public system, we have 2 specialist cancer genetic centres that accept referrals from all around the country. And these services are experiencing sustained pressure due to the increased demand for cancer genetics. And this demand is due to increased knowledge about the role of genetics in tumour development and the need for us to know this information in decision making. But there's also a greater appetite amongst the public who have had a personal or family history of cancer to know this information to guide their own care and the care of their families. We've also seen huge advances in our genomic technology and what we're able to test, and an expansion in the criteria for testing, so more people are eligible for testing. And this growth, this demand is anticipated to continue as genetics becomes a core component of routine cancer care. But what we have created is this bottleneck scenario where the services we do have are inundated from referrals from our regional cancer centres, our surgeons who need this information, and our GPs who are facing these questions from the public. And it is not uncommon for people to wait over a year and sometimes 2 years to access these specialist cancer genetic services. We also face some challenges locally. There can sometimes be uncertainty around referral criteria and a variation in the understanding of eligibility, and this can lead to over and under referral to specialist centres. And there can be a lack of clarity on the referral pathways, where the referral should be initiated, should it be at diagnosis or when in oncology. And because of the extensive wait times, it's having a real impact on time-sensitive decision-making in day-to-day practice. It's sometimes the case that by the time patients are seen by the specialist cancer genetic centres, the referrer is no longer actively managing that patient, and there can be some ambiguity on who should act on the recommendations if a diagnosis is made. We sometimes see inequitable access to existing services because, of course, there are many routes to private assessment and testing in Ireland if patients have private healthcare or are able to afford to go down this route. And sometimes we have the scenario where patients wait so long for assessment they no longer see the importance of it and they may not attend and they could be lost to follow up. Or we have the other scenario where they may bypass the system altogether and go down the route of direct-to-consumer testing, the likes of colour that can be bought online. And this can really lead to lack of clarity around the test results, especially in the absence of a genetic counsellor or a multidisciplinary team. So bearing in mind these challenges, it's very clear that a structured approach was needed in order to provide an efficient service. So back in 2022, the HSE's National Cancer Control Programme published the National Strategy for the Acceleration of Genetics and Genomics in Ireland. And this was a huge body of work that really recognised the great work that was being done and kind of made a commitment to grow and build on this. And the aim was really to provide this structured approach to the development of genetic services to build the workforce of the future and to provide the infrastructure so that we were able for the demand that was coming our way. And this led on to the hereditary cancer model of care, which was published in April 2023, which really outlined how cancer genetics can be better integrated into our existing services to use the expertise that we already have in our centres to identify, assess and test those who with a suspected inherent predisposition to cancer. And therefore relieve that bottleneck that we spoke about so that we are only referring on those with a positive test results or those with maybe a severe phenotype or a variant of uncertain significance in a high-risk gene. And all this will be overseen with direct referral pathways and coordination at a national level. And St. Vincent's would have been the first hospital to pilot this approach and have been hugely successful in doing so. So we are seeing many other centres across the country take this approach. Cork have got up and running, and now we are doing the same, trying to get started here in Limerick. And this is just a snapshot of what the regional services will look like. So it's a hub and spoke type model. So locally we will have our team that will link very closely with the specialist cancer genetics team nationally. So in Limerick we're very lucky. One of our oncologists, Dr. Aleem, has taken over the role of clinical lead of cancer genetics, and we've great support from our surgeons, from our oncologist. I have gone into post as a colorectal AMP, and hopefully in time I'll see counterparts in gynecology, breast, haematology and general medical oncology. We have admin support, and we link very closely then with the special cancer genetics team and have direct access to a genetic counsellor for our patients who undergo mainstreaming. We link in with National MDT and have access to a genomic research associate through this MDT. In terms of the hereditary colorectal clinic, the aim of this clinic is to have full involvement in the patient pathway and to be that point of referral locally. If there is a patient with who's suspected of having an inherited predisposition, they can be referred to this clinic and undergo comprehensive personal and family history assessment. They can be assessed against criteria for testing, and if eligible, can undergo pre-test counselling and consent locally. And the appropriate germline testing can be taken place all under the one roof, with timely and direct access to a genetic counsellor for those with a positive test result. But this clinic will also offer continuity of care, coordination of referrals and follow-up of this patient group. So when we're talking about inherited hereditary colorectal predispositions, this slide will summarise what we're talking about, and we could probably talk all day about this slide, but we don't have the time. But we can divide these into our non-polyposis and our polyposis conditions. So Lynch syndrome there, of course, being the most common inherited colorectal predisposition affecting approximately 1 in 270 people. And we have our mismatch repair genes there. In Lynch syndrome, we see a deficiency in one or more of these mismatch repair genes. But we also have our predispositions that would be associated with a polyp burden. We have our adenomatosis, our serrated, our hamartomas, and our mixed polyposis group. And we have our FAPs and our MAPs inside there as well, our Peutz-Jeghers and our juvenile polyposis syndrome, but again, many rarer conditions there as well. And the genes that we see here would be the genes that we would test in a typical hereditary colorectal panel. So where to start in terms of service development? So we've started with the cancers, basically. So we are ensuring that everyone who is diagnosed with a colorectal cancer in UHL undergoes the Lynch syndrome diagnostic pathway. And this is basically where they will have tumour-based mismatch repair testing on the first available sample that we have. And this is as recommended by the NICE guidelines since 2017. And as Prof. Lemming said, we were probably not doing this very well until recently, and I'll talk about that in the next slide. But very importantly, having access to germline testing for our early onset colorectal cancer, as we've heard about in the last two talks, will be hugely important, and being able to access that in a timely fashion would be a priority for this clinic as well. But over time, it's hoped that the service will include our patients with polyposis, those with family history, and that we can facilitate cascade testing for family members. So the Lynch syndrome diagnostic pathway is basically, it's a cascade of tests that needs to be followed for our patients who are diagnosed with colorectal cancer. And it starts with immunohistochemical testing of the tumour for the mismatch repair genes. And depending on those results, there's further tests that can be ordered when we see a loss of MLH1. And I'll just show on the next slide. So I went into post in February, so we've started recording this in a very systematic way and capturing the data. So we'll say from the end of February to the end of March, at our colorectal GI MDT, 34 new cancers were discussed. They all underwent MMR testing. Of those, 28 were proficient for the MMR proteins, but 6 were deficient. So those 6 required further testing. All 6 of those showed loss of MLH1 and PMS2. And in that scenario, we need to test for BRAF. So all 6 had BRAF testing, and 3 of those were shown to have the BRAF V600 variant. So when we see BRAF in the presence of deficient MMR cancers, we don't associate that with Lynch syndrome. And so no further testing was required. But the 3 that did not have the BRAF variant required further testing. They required MLH1 promoter hypermethylation. This was performed. 2 did not, or 2 had hypermethylation. And again, we call this explained mismatch repair deficiency. So we don't associate that with Lynch syndrome. So the one lady that had promoter came back as unmethylated. She requires germline testing. And I always say that this just takes into account the tumour. We always have to take into consideration the patient that's in front of us in the clinical presentation. So we'll continue to record this data. And, you know, the first month of this, 53% of this was reflexive, 47% of this had to be requested. So the hope is that this will become automatic routine, and I already see the improvement in that. So the benefits of providing this service in a timely manner locally allows us to be proactive about the management of our patients and to implement surveillance and risk reduction strategies. It also gives us information about predictive and prognostic treatment planning, what is likely to help our patients. We see huge benefits of the use of immunotherapy in our mismatch repair deficient colorectal cancers. It allows us to have these really important discussions with our patients in terms of, you know, the likes of family planning, facilitating that discussion and referring onwards. With our younger population, who there is the option in Ireland of pre-implantation genetic testing. While it's not publicly funded and is probably a long way off being publicly funded, it's a conversation that we can initiate and refer onwards. It'll also allow us to perform appropriate cascade testing of family members and implement chemopreventative strategies. So I'm talking about the likes of aspirin in our Lynch syndrome population. We have 2 important pieces of evidence there from the CAP2 and the CAP3 study that provides us with the information that the use of aspirin can reduce the risk of colorectal cancer in this high-risk population group. So when a diagnosis is made, it's really important that we're using correct, up-to-date guidelines in terms of management and surveillance and risk reduction. So for our Lynch syndrome patients, we have the Monaghan paper, which is a combination of British and English or Irish guidelines, and they tie in with the European guidelines from the European Hereditary Tumour Group. And these are summarised excellently on the UK Cancer Genetics Group that gives us fact sheets, gene-specific, gender and age-specific guidelines. For our polyposis, our patients with polyposis syndromes, we have the Zaffaroni paper, which our very own Professor Fleming was a contributor and co-author on this paper that gives us the guidelines on how we manage these rare polyposis groups. And again, for the Putzjäger, we have our Wagner paper there. There is a group of patients, approximately 15 to 20% of patients that will have a family history, but will not have any pathogenic variant after they go through germline testing. And it's really important that this group of patients will receive specific guidelines on their management also. And it's the hope that the Hereditary Colorectal Clinic will be able to give that information and stratify them based on average, moderate or high risk. And the Monaghan paper will help us to do that. And beyond diagnosis, it's really important that this hereditary colorectal clinic will provide ongoing care for patients in the Midwest, and that will follow patients who have a hereditary predisposition to ensure adherence and surveillance and compliance with prevention if they're on such, and maybe assess why that might not be happening, and to check in on psychological well-being. As life evolves, so does the coping. Maybe someone in the family has been affected by cancer, and these patients may need to link back in with our psych-oncology services. Again, allows us to have those important conversations about reproductive and life planning that might not have been important at the time of diagnosis, but can be later in life. And to have those conversations about the cascade testing and how to approach conversations with family members, to deal with the social and practical issues around having a hereditary predisposition to cancer, insurance, employment rights, using those appointments as a point for health promotion and lifestyle risk modification, and checking in on the guidelines that were originally given to make sure that they are still up to date, and it's a dynamic field that maybe these guidelines have been modified since diagnosis, just to check in on that. And to signpost to the very important resources that we do have for our patients who have a hereditary predisposition to colorectal cancer, and just provide that support. Thank you.
So our next session is looking at risk factors and health in cancer, and it has a bit of a focus on metabolism, nutrition and different nutrients. And I think it's going to be an interesting session because it's going to show, even though we're focusing on a particular theme, throughout the LDCRC we're doing it in lots of different ways, whether that be clinical, cellular or physiology. And first up is Dr. John Noon, who's Assistant Professor of Exercise Physiology in the Department of Physical Education and Sports Sciences. Dr. Noon completed his PhD at Dublin City University, and him and I just were passing ships in the night. I went into DCU, into the NICB as he left, and I was delighted to see him come back to UL as well. And he's going to talk today about his work on the role of the endocannabinoid receptor signalling in skeletal muscle degeneration during cancer cachexia. Thanks very much.
Okay. My name's John Noon. I'm an assistant professor in sport and exercise physiology at the Department of Physical Education and Sports Sciences, so thanks for having me. So cachexia is a condition that impacts many individuals who suffer from cancer. Almost 60% of individuals who suffer from cancer, specifically GI and lung cancers, suffer from cancer cachexia. And it's a condition whereby we see significant declines in skeletal muscle mass and in adipose tissue. So as a result, it requires substantial research. What we see is that those individuals who suffer from this condition have declines in physical function and quality of life. We also see that there's major declines in survival, and the treatment response is very, very limited and very low. So as a result, we need to kind of look to develop alternative therapies. At present, there's no real major approved therapies for the cancer cachexia, whereby we have trials looking at exercise resistance and endurance exercise in particular, but of course it's a challenge to ask individuals who are undergoing cancer therapies to participate in exercise, particularly strenuous exercise, in order to limit the changes in muscle. Furthermore, and I'm not the specialist in this, but our next speaker will have more details on this in respect to nutrition, but there are nutritional methods that are being used in order to limit the changes that we see in muscle and in adipose tissue, particularly high protein diets focused on branched chain amino acid supplementation. And there are some pharmacotherapies, for example, corticosteroids, in order to increase appetite and decrease inflammation in these individuals. Now, the methods for its assessment are quite limited, but also varied. So we see that, as you'd expect, we have clinical measurements, for example, anthropometrical measurements, weight loss, strength changes, changes in muscle mass, body composition, but we also have our molecular assays, which we're all familiar with, gene expression, proteome, lipidome, metabolomic, etc. But we also have our experimental models to study muscle loss, for example, cell culture, tumour-bearing mice, etc. But one area that we have shown some kind of progress is that, bears, interestingly enough, they actually uniquely preserve muscle mass in incidences where they go through prolonged physical inactivity. We see that bears in the wild, they tend to maintain muscle mass after prolonged physical inactivity, as I said. This is quite unlike us as humans. When we go through prolonged inactivity, we actually lose muscle mass. So we kind of use this as maybe a model that we could use to assess or to see whether or not there might be mechanisms in which bears maybe maintain muscle mass and how that can be applicable, for example, in cancer-cachexia conditions, which is something that we have now tried to do. So what we have seen is that, interestingly, a specific system within the skeletal muscle called the endocannabinoid system, which is involved in many factors, many factors from mood to inflammation to metabolism, is actually changing in response to hibernation. And in fact, if we focus on these two specific markers, which is the endocannabinoid receptor 1 and the endocannabinoid receptor 2, we note that the cannabinoid receptor 1 is actually significantly changing or declines after hibernation. But interestingly enough, we don't see any significant changes in the endocannabinoid receptor 2. So it kind of identifies maybe there might be an avenue that we could go down within maybe human models that we could explore. Well, why is how could we kind of manipulate this and use this to study from our perspectives? So we really wanted to see whether or not the endocannabinoid system maybe has a role in controlling in muscle wasting conditions. So this is something that we've done. Myself and my colleagues in Trinity have looked into the development of an in vitro model system for cancer-cachexia, where we looked at both the SW480 and H1299 condition media. So we grew these human colon cancer cells and these lung cancer cells, and we grew them in vitro, and we extracted the condition media from them. And we measured many markers, many cytokines. In fact, we found that there was predominant changes in particularly interleukin-1, IL-6, IL-8. These are factors that are highly expressed in cancer-cachexia. But interestingly enough, this one that I have highlighted in bold was really highly expressed. This is TNF-alpha, and quite interestingly enough, this has been originally identified as being cachexin. So cachexin was the main protagonist for cachexia, was identified as being, or originally was thought to be, the main protagonist for cachexia. So this was kind of a nice thing to identify, that all these cytokines were highly expressed within our conditioned media. So we wanted to see whether or not we could expose this then to our human skeletal muscle cells. So we grew human skeletal muscle cells, and this is something that we do quite frequently here in UL. And we exposed our conditioned media to our skeletal muscle cells to assess to see whether or not there were changes in the muscle when they were exposed to this conditioned media. And what we found, if you can follow this slide, is that with different concentrations of conditioned media, be that the GI specific conditioned media or the lung specific or lung cancer cell specific media, we showed that there were significant declines in myotube diameter or muscle cell diameter when they're exposed to higher concentrations from 30%. So this is evident here in the blue. We see that there's significant declines after 30%, and also in the green here after 30%. We also observed that those conditions that were greater than 30% evidenced significant cell death. So essentially, this was aligned to what was shown by Seto and Jackman, who emphasised that in a mouse skeletal muscle model, that there were significant declines and in myotube diameter at 30% conditioned media, and it was reflective of cancer-cachexia in vivo. And this is something that we then have now replicated in human skeletal muscle cells, which is quite exciting. So the next thing then we wanted to do was to assess whether there was a role of the endocannabinoid systems in inhibiting or activating any of these particular changes that we see in a cachexic environment in vitro. So what we did was we looked at an endocannabinoid receptor agonist known as THC that activates both cannabinoid receptors 1 and 2. And what we showed was that when we exposed the cells to endocannabinoid receptors, THC, we observed both in the colon cancer condition and in the lung cancer condition that there was no significant declines in myotube diameter, in both conditions. So activating the cannabinoid receptors 1 and 2 led to a maintenance in myotube diameter. We then further wanted to see, well, because of this bear model, and they showed that there was no changes in this cannabinoid receptor 2, well, what would occur if we looked at specifically at endocannabinoid receptor 2 and activating it with JWH133? And what we found was that when we exposed the cells to conditioned media and JWH133, there was no significant decline in myotube diameter in both the colon cancer condition and in the lung cancer condition, which was quite exciting. We then looked at a specific drug that was developed by Artello Biosciences called ARC27.13. And what we showed, which is a cannabinoid receptor agonist of type 1 and type 2. And what we found was that when we exposed the cells to the GI condition and the lung condition and the ARC27.13, that it maintained myotube diameter in both conditions. The next thing really, however, it doesn't really tell us anything mechanistically, unfortunately. So we wanted to see whether if we blocked endocannabinoid receptor 1 or 2 and activated either 1 or 2, well, what was the change? So we did that. So what we did, we activated with ARC27.13, we activated the cannabinoid receptors 1 and 2, and then blocked with an antagonist with using rimonabant, which is a cannabinoid receptor 1 antagonist, and then AM630, which is an endocannabinoid receptor 2 antagonist, and look at the effects. And what we see here in this green line is the changes in ARC27.13. It maintained myotube diameter in both conditions. However, when we blocked endocannabinoid receptor, when we blocked the endocannabinoid receptor with rimonabant, which is an endocannabinoid receptor 1 antagonist, we showed that there was no change in myotube diameter either. This is similar in the lung cancer condition. In the lung cancer condition. And when we blocked, activated the cells with endocannabinoid receptor 1 and 2 with ARC27.13, and then blocked the cells with cannabinoid receptors 2, we showed declines in myotube diameter, suggesting that the changes that we see driven here are potentially driven by the endocannabinoid receptor 2. So overall, activating cannabinoid receptor 1 and 2 and blocking cannabinoid receptor 1 actually maintained myotube diameter in cachexia conditions. Activating cannabinoid receptors 1 and 2 and blocking the cannabinoid receptor 2 actually led to myotube diameter changes and declinations in cachexia conditions. And then maintenance of human skeletal muscle myotube diameter may be actually driven by this cannabinoid receptor 2 agonism in cachexia conditions, which is something that we actually potentially saw originally in this bear population, which is quite nice to see that translation. So this is quite nice, as this work has now gone on to clinical trial in phase 2, which is ran by the CARE study, and it's shown to have a positive effect in weight gain of 6%, in comparison to an average weight loss in those individuals who didn't take the drug of 5.4%. So showing that there may be a potential effect of weight gain in people who take this over 12 weeks. So overall, there's preclinical evidence to support the role for cannabinoid receptor 2 agonism under cachexia conditions in vitro. Clinical observations bodes well. However, there's much more work to do within the clinical trial that's been done with the CARE study. And the mechanisms of action potentially are through this cannabinoid receptor 2 agonism, but there's certainly more work to look through the potential alternative mechanisms that may be working in sync with cannabinoid receptor 2 agonism. So thanks very much. These are my acknowledgements, and thanks for having me, and I appreciate having me here today. Thank you.
Thank you very much, John. So next up we have Assistant Professor Clodagh Scannell. So Clodagh is a core registered dietitian and has recently joined UL and the LDCRC as Assistant Professor in Human Nutrition and Dietetics. So she has a lot of interest in clinical nutrition in oncology, looking at the effect of body composition phenotype on things like cancer cachexia, like John introduced, and also sarcopenia. And Clodagh is also the co-author of a book called The Truth Behind Food and Cancer, which is a freely available educational resource for patients that are diagnosed with cancer. So thank you very much, and best luck with your talk.
So hi everyone, my name is Clodagh. I'm a registered dietitian and I'm new to the School of Allied Health, so I'm delighted to be here for my first research week. I'm going to talk about some of the work that I conducted during my PhD in UCC with the hope that it might form some collaborations within the LDCRC. So my work primarily focuses on cancer-associated malnutrition, and this form of malnutrition is present in 31 to 87% of patients, depending on tumour type, stage of disease. And it's been shown time and time again that it negatively impacts on someone's quality of life, how they tolerate their treatment, and their overall survival. But malnutrition with regard to cancer, it's a lot more complex than just weight loss. It also encompasses an array of abnormal body composition phenotypes, such as your sarcopenia, your mycosis, your frailty, and as John alluded to, the whole trajectory of cancer cachexia. But on the wards at the moment, and from what I'm seeing myself, is that patients are no longer presenting as like the typical frail cachectic phenotype that we're used to. And it's been estimated that about 60% of patients, even in those with metastatic disease, are overweight or obese. And this makes it difficult for us as dietitians and other healthcare professionals to identify those who have malnutrition, because it can be hidden beneath the surface of the skin and beneath excess adiposity. And in this instance, I suppose traditional anthropometric measurements such as taking someone's weight, calculating their BMI, their body surface area, it essentially tells us nothing about body composition. So we have this really unique opportunity in oncology nutrition, particularly oncology research, where we can use routine clinical or CT scans that are taken for the purpose of screening, diagnostics, treatment planning, to perform body composition analysis. And this is considered the de facto gold standard of body composition analysis. So if you take a cross-sectional slice at the L3 vertebra using equations and software, this allows you to precisely quantify whole body skeletal muscle and adipose tissue. And Christina kind of had a slide about this earlier, but essentially this is an example of one of the images that we would see. And you can see that different areas light up according to density. So you have the blue colouring, which is the subcutaneous fat directly beneath the surface of the skin. Then the green colouring is your visceral fat, so fat that's in around your organs. And then the red is the muscle, which is something that we're most particularly interested in. And usually you'd like to see this like red, thick band around the whole circumference of the stomach. But you can see that that's not the case there, and you have these specks of yellow, specks of blue. And this is fatty infiltration of the muscle. So that would indicate to me and to others that this is poor quality muscle. So just to further build on the point that malnutrition influences survival, we conducted a study where we collected clinical data from 1,405 participants, including CT scans. And it was data that was collected from 18 clinical sites across Ireland and the UK. It was a mixed bag in terms of cancer diagnosis, cancer stages, treatments, intent. But regardless of this, we found that about 40% of our cohort were diagnosed with malnutrition according to GLIM. So you don't need to know anything about GLIM, it's just a framework how we as dietitians diagnose malnutrition. And it splits it into moderate malnutrition or severe malnutrition. So 40% of our mixed cohort had malnutrition according to this framework. So then we performed a multivariate survival analysis, and survival was followed for about 8.5 years. And what we found was that there was a statistically significant difference in survival depending on whether you had malnutrition or not. So then, to kind of follow on from this, I suppose this is something that's very present at the moment, and it's something that's definitely contributing to cancer-associated malnutrition. And it's something I'm particularly interested in. It's the widespread prevalence of misinformation and disinformation. And it's been shown post-cancer diagnosis, patients are very vulnerable, but they also have this increased motivation to seek out nutrition-related information. And it's been estimated by the Irish Nutrition and Dietetic Institute that there's only one dietitian for every 4,500 cancer survivors in Ireland. So what this creates is this huge gap where patients are eager and keen to find out information about diet, but there's not enough qualified individuals to provide that advice. So what happens is they go online, and you can see this is what I've done, and nutrition and diet, Google diet and cancer search, and these are all the titles that pop up, and you can see they're quite eye-catching in How Not to Die cookbook, the cancer prevention diet, foods that combat cancer. And what this results in is patients with potentially poor health literacy, or those just looking for a silver bullet, initiating some special diet, some fad diet, some practice, some products that's contributing to cancer-associated malnutrition. So we were particularly interested in this. So just to give it its definition, we call these practices and these products biologically based complementary and alternative medicine. So this includes special diets, dietary supplements, herbal remedies, botanicals that are not prescribed by a doctor or a dietitian. So for the most part, maybe potentially some of these are harmless. albeit expensive. But there is the situation where some of these products, these practices can cause harm. So I think what a lot of people forget, particularly with minerals, vitamins, herbs, is that they are biologically active and they have the potential to interact with conventional cancer treatment. So, for example, they could make someone's chemotherapy less effective or potentially more toxic than originally planned, and you're ending up with maybe debilitating side effects. And then, as I've already alluded to, these special diets, they are largely restriction diets, exclusion diets. So they result in people getting less calories and less protein than required. And they're not recommended by any of our evidence-based guidelines. And then, as well, what we've seen is that there's lots of concern around the safety and the legality of some of these products. They're being bought online and they're not undergoing the same rigorous testing that they would to get on the shelves here in Ireland. So, as I said, we were very interested in this. We conducted a national survey to find out, well, what are patients here in Ireland doing? And this is sponsored by the Irish Cancer Society. So in our about 360 respondents, we found that about 1/3 were engaging with biologically based complementary and alternative medicine, and that the use increased significantly post-cancer diagnosis. So people were taking high doses of minerals and vitamins, dietary supplements, herbal remedies, things like cannabis, vitamin B17, which is like an artificial vitamin, even IV vitamins. And then in terms of special diets, which obviously is relevant to us as dietitians, dairy-free diet, gluten-free diet, fasting, ketogenic diet, detox diets. The patients reported that they were taking these products because of symptoms like fatigue, disturbed sleep, a fear of recurrence. And then in terms of perceived benefits, they reported that it improved their overall well-being, it reduced their psychological stress, and they felt that they did improve their quality of life. And then, as I kind of mentioned, we found that the majority of our cohort were using the internet to find out information about biologically based complementary and alternative medicine use. So I suppose this kind of got the cogs turning, and although there was not much we could do about dietetic staffing levels in Ireland, we did think, well, okay, there's this huge gap for evidence-based resources. So what we did was we used the information that we got from the survey about all the weird, wacky things that people were doing, and we decided to write an evidence-based scientific rebuttal that was layperson-friendly. It was written by a team of dietitians in UCC alongside a consultant medical oncologist. We were then endorsed by the Irish Nutrition and Dietetic Institute, the Irish Society of Medical Oncology, and the National Cancer Control Programme here in Ireland. We had patient representatives involved at all time points, and then we had a graphic designer jazz it all up and make it look nice. And then we got into talks with Breakthrough Cancer Research, who could really see the value of what we were trying to do. And they very kindly sponsored the project. And we had 20,000 copies printed, and they were freely distributed to cancer centres nationally, and it's also hosted as a PDF on their website, just to be available for patients at the source. So then, just to conclude, we know that cancer-associated malnutrition is highly prevalent, and it's associated with detrimental consequences such as reduced survival. Malnutrition can look very different on different patients, and we need to consider body composition. And then there's kind of the cascade of increased motivation post-diagnosis with unmet needs, gaps in services, heightened use of the internet, and this increases the use of fad diets and unregulated products. We probably need to remind patients that something that's natural is not harmless. And then the truth behind food and cancer is just an example of translating evidence into practice. Thank you very much.
Thank you so much, Clodagh. So last up, we have Caitlin O'Grady. And I'm really delighted to introduce Caitlin here as representing our early career researchers and our PhD researchers as part of the LDCRC, who are usually the ones doing all of the, work at the coalface in the lab day after day. So Caitlin's going to talk about her work on 3D models of prostate cancer, which she uses to investigate zinc homeostasis and zinc-regulated cellular processes.
Hello everyone, my name is Caitlin O'Grady and I'm a PhD student here in the Department of Biological Sciences in Brennan Institute in UL. And I'm just going to talk a little bit about my PhD research here today. So I'm going to talk a bit about zinc and how it affects prostate cancer biology. So just a quick introduction first. So prostate cancer is a common and growing challenge despite advances in different treatments. It still remains a significant clinical challenge, as it affects 1 in 8 males. They will get prostate cancer in their lifetimes. I suppose the main function of the prostate as well is the secretion of prostatic fluid, and prostatic fluid is really high in citrate and zinc. And this, we'll say another interesting thing about the prostate is that it has the ability to accumulate lots of zinc, whereas in cancer, it loses its ability to accumulate zinc, and this is through the loss of the main zinc importer, which is the ZIP1 importer. So this reduced amount of zinc in cancerous tissue is actually a hallmark of malignancy. So why zinc? Why is zinc really important? So in healthy tissue, like I said, it's able to accumulate lots of zinc, and this is really important because it's able to inhibit the TCA cycle. And the TCA cycle, when the zinc inhibits the TCA cycle, this reduces the ATP production, and then the low metabolic state of the prostate is conserved then. Whereas in prostate cancer, the TCA cycle continues, it's active, and then this increases ATP production, and this supports all of the ATP that's produced. This supports cancer cell growth then. So I suppose the main hypothesis then of my PhD is if I can restore this intracellular zinc levels, this could be a potential therapeutic strategy then. So this is one of the challenges as well. Zinc supplementation is a great idea, but there's not all zinc supplements then are created equal. So unfortunately, most of the zinc supplements that are on the market at the moment contain inorganic zinc sources, and these face poor absorption, whereas so we have a collaborator in America called Zinpro, and they specialise in zinc supplements as well. But they specialise in zinc supplements for the farming industry. So their zinc supplements are different. They're zinc amino acid chelates. So these zinc amino acid chelates get absorbed through a different pathway. So yeah, on the left-hand side here represents my inorganic zinc sulfate, which is was commonly in zinc supplements on the market. And these face competition. So the zinc will face competition with other divalent metals such as iron and copper, whereas the advantage then of the zinc amino acid chelates is that they get uptaken through a different pathway. So they get uptaken through amino acid transporters. So it's kind of like a Trojan horse effect, and we'll say the membrane, cell membrane sees the amino acids, and it's like, okay, you're coming over here, so that the zinc gets uptaken a lot better. So then I suppose my research focus then, the main question that I'm looking at is, can we restore the low metabolic state with extra zinc in the cancer cells? So first of all, I'm going to look at metabolism. So I want to see if zinc alters mitochondrial activity and overall metabolic function. And then also I have grown 3D tumour spheroids out of prostate cancer cell lines in the lab, and I want to see if this, we'll say in a more 3D biomimetic environment, how zinc influences cancer cell survival in this model. And then obviously I'm going to see my 2 different formulations of zinc. So I'm going to use my inorganic zinc sulfate and then my zinc amino acids as well to see if there's any difference. So first of all, I did a seahorse mito stress test just to see if my zinc alters mitochondrial metabolism in the prostate cancer cells. So this is my first graph. So my control is in blue, and my zinc sulfate is in red, and my zinc amino acid chelates are in green. So this is just a measure of mitochondrial respiration. So as you can see, the zinc amino acid chelates reduces metabolic respiration compared to zinc sulfate, as well as my control. And this indicates a stronger suppression of mitochondrial respiration. But then also ultimately this suggests that there's improved intracellular delivery of the zinc with the amino acid chelates versus my inorganic zinc sulfate. And then we'll say this gets into the mitochondria as well because it has this nice desired effect. So then next I have grown 3D tumour spheroids as well in the lab. And I wanted to see if these, we'll say in a more 3D environment, if it would have the same desired effect. So here I have my control. So these are 3D tumour spheroids made from PC3 cells. And I've done a live dead staining here. So the green is live and red is dead, and then the merge is just an overlay at the end. So my next control then is my amino acid control. So these spheroids have been treated with the exact same concentration of amino acids that are in my amino acid chelates. And they look very similar. Then my zinc sulfate here has been treated with the exact same concentration of zinc as my zinc amino acid chelates. The only difference is that the amino acid chelates they're chelated to amino acids. And then here are my zinc amino acid chelates. It's probably a bit hard to see there. But I think what's really nice about this is that it shows that between my two controls and my zinc sulfate, they're all very similar. Whereas when you see the zinc amino acid chelates, there's a really big difference. And then this shows that the zinc amino acids induce metabolic collapse rather than maybe dysfunction at this time. And then because the zinc is able to get in, it's delivered better. Then the ATP has been stopped producing. So then this leads to energy starvation for the cells. And then this leads to reduced viability then of these tumour spheroids. So then just some conclusions. So I suppose the question I started off with, does zinc change prostate cancer biology? I think after studying it for the last few years of my PhD, I think it definitely does. So I think, hope I've convinced you of this as well. Zinc formulation affects cellular uptake. So you can see the difference between my inorganic zinc and my zinc amino acid chelates as well. And then from my seahorse mito stress test, you can see that the zinc induces metabolic disruption in the prostate cancer cells. And then zinc reduces my viability in my tumour spheroid model as well. So hopefully all of this kind of supports the idea for a development of novel metabolic-based therapies. I think it's all looks very promising, but I'd also like to look at some like future work for me. I'd also like to look at some other targeted delivery systems. So I'd like to look at developing targeted zinc-loaded nanoparticles. This would allow me to kind of more target prostate cancer cells, and then and then I've also seen a paper recently as well that zinc actually increases chemosensitivity. So just a nod to Claude there from the last talk. So even to co-encapsulate the zinc with some chemotherapeutics as well. And then to validate the system then in my 3D tumour spheroid model. And then I'd also like to look at if zinc influences invasiveness in 3D spheroids by EMD changes as well. So just my acknowledgements. I'd like to thank all my supervisors, the Doctoral College here in UL and Research Ireland for funding my PhD, my collaborators as in Pro in America, and I'd like to thank everyone from the biolabs in Brennan. You're all amazing to work with every day. And thank you all for your time as well.
Okay, thank you very much. Thanks for staying this long, and to all our speakers to date. I'll quickly highlight some of the clinical research and clinical trials and advertise a few groups that we're looking for to recruit into. So I'm not looking to end on a bad note. There are lots of improvements in the Midwest, and this data that I'm showing of the poor outcomes for cancer patients in the Midwest compared to the national average come from a number of years ago. There are many reasons for this. A lot of our rapid access programs were not rolled out at the same time as Dublin rapid access programs, so some of it is all corrected now, but I just want to highlight that we still have to do better. And if we work in a research centre, so if our cancer centre is research active, we know from international best practice that we have better outcomes for our patients. It's just to highlight that fact today, but we know this already. And the other point I wanted to make was we're looking to get accreditation for our cancer centre, and even though we have two institutions, we work as one across UL and UHL for cancer research, and we are embarking on an accreditation process looking to raise the standards for our cancer centre in line with best practice across Europe. So you'll be hearing more about this, but just to use this opportunity to highlight this OECI process that we're undertaking, and many people in the room are helping us with this. So as you can see here, our vision in Limerick Cancer Trials is to deliver innovative interdisciplinary clinical trials. So we're not just looking at drug trials, we're looking at other types of trials that can improve patients' outcomes. And we're looking across various themes that you can see highlighted here that are future-proofing our clinical trial strategy. You've seen some of these slides this morning if you were here, as I collaborated with Pat on this, just to highlight the people who are involved in leading these various work packages to make sure we have holistic types of clinical trials, not exclusive clinical trials, that we're addressing inequalities, and then digital innovations through the LDCRC underpinned by excellence in education and training. And this is our Limerick Cancer Trials team. We're based in UHL, so our Cancer Clinical Trial Unit. And you can see we have a wide variety of PIs, and this team is very much focused on drug interventional studies. And our drug interventional studies are increasing, delivered by this excellent team to a very high standard. And this is really just taking off in the last couple of years because we have protected time for PIs and we have an increased number of PIs and investment as well from our HRB grant. And I just want to highlight all the collaborators and networks from our local PIs in UHL and UL to national partners that support us to improve the number of trials, but the number of patients coming onto clinical trials. Cancer Trials Ireland is a key network in Ireland. This is a charity that aims to maximise the number of cancer trials, and we very much run multi-centre, multi-site trials led out and sponsored by Cancer Trials Ireland. We've a lot of international clinical trials from international cooperative groups that are led out through Cancer Trials Ireland, and we also have a really nice project led out by myself and Patrick with Cancer Trials Ireland on equitable access to clinical trials for our cancer patients, leading on the Midwest, linking with what Christina was presenting earlier. And finally then, I just want to highlight that there are different types of clinical trials. We have focused over the last years, focused our strategy on drug interventional studies, but there are many other studies that benefit patients. Clodagh was involved in a big study, the Lee Mack study down in Cork, looking at dietary and exercise interventions. And we're very interested in this now, moving on to sort of phase 2 of our strategy. And we're undertaking workshops to bring in more PIs. So this is a little bit of an advertisement. If you're interested in cancer interventional studies, Cancer Trials Ireland are very much supporting us. And please feel free to link in with us, with Tricia or Jenny, with the LDCRC or Limerick Cancer Trials to join any of our subgroups, or indeed open up a new subgroup. We meet monthly or less frequently, depending on the subgroup, where we discuss our clinical research, our translational research, our biobanking. We bring together ideas, we bring together collaborators, PIs, look at what resources are needed. Just feel free to link in if you want to join in. And just to highlight our patients, we have a very active PPI group running for 4 years now. We have 17 members in our PPI group, and then our PPI steering committee, many people in the room are involved in that, UL, UHL and patients. And this helps shape our strategy. It helps, these patients help review our research documents, help us plan grant submissions, and we're very proud of this group. And we're the first hospital-based PPI cancer research group, and we're just growing in expertise. So just to say that recently we have 12 principal investigators leading out on 10 investigator-initiated studies. So these are going to be our non-drug interventional studies that are focused on various themes. You can see some of the themes there down the bottom, just to give you an idea of what we can grow from our centre in Limerick. It's not that we can't design our own drug interventional studies, but that comes at great cost. So we're looking to begin with this strategy, and of course we can develop drug interventional studies at a later time. So we have our next meeting of the LDCRC and Limerick Cancer Trials on Friday afternoon, the 19th of June. And during that meeting, we will further develop some of these investigator-initiated study proposals. So feel free to attend and bring your ideas. All are welcome. Thank you very much. I'm going to... So with that, I'm going to hand over to Aideen to close the meeting, and just to thank you again today for all of your support, enthusiasm, and to our speakers for a great afternoon. Thank you very much.
Okay, right. I am incredibly grateful to everybody for coming today. And as you've seen, like today, we very much focused, I think, the day on diet and exercise, and like it's been different flavour to some other meetings. And as we move through, we hope to really represent all of the different aspects of cancer research going on in the Midwest. But I really want to highlight a couple of upcoming events that we're incredibly excited about. So on the 6th of May, we will have, which is really soon, it's next week. Next Wednesday. Oh my God, we're into May already. So just straight after the bank holiday weekend, Professor Helena Vasconcelos, and I'm probably pronouncing that wrong, from the University of Porto, will be here visiting, and she's very eager to undertake a sabbatical visit or a professor visit here. So she's eager to engage in collaboration. She looks at exosomes, ctDNA, and drug resistance, with particular interest in lung cancer as well. We're incredibly excited. On the 14th of May, we have a public event. This is for the public to come in. We will be inviting 100 members of the public to come and visit the labs and see what actually happens in cancer research labs. And we have multiple stations in that. And Amira is doing an incredible job on leading out with Tricia and the team on organising that event. It's an evening event from 6 to 8 p.m. The Eventbrite is open. If you have, family or neighbours that you think that might be interested in learning about cancer research, please invite them to come. It will be sponsored by Breakthrough Cancer, and they do a phenomenal video that they share on their social media, which will actually hopefully get the news out about our centre. And finally, as Ruth mentioned, in June we have the next meeting. So thank you so much. Thanks everybody.